So we know that the metabotropic glutamate receptor 3 is involved in human cognition. And we were interested to see if N-acetyl-aspartyl-glutamate, or NAG, which is the endogenous substrate for mGluR3, was associated with cognition also in human brain. So we studied the enzyme FOLH1, folate hydrolase 1, which codes for the enzyme called glutamate carboxypeptidase 2, or GCP2, which is responsible for breaking down NAG in human brain. And we looked at the relationship between FOLH1 and GCP2 and NAG and its role in cognition in humans. We did this in both healthy subjects and psychotic patients, including patients with schizophrenia. So we found that a missense mutation in FOLH1 was associated with increased expression of FOLH1 in the prefrontal cortex of patients with schizophrenia and healthy controls. And we found that other patients who had that mutation associated with higher levels of FOLH1 had lower NAG levels in human brain. And we found that patients who had lower NAG levels in their brains had poorer cognition, specifically poor visual memory and working memory. So carriers of the FOLH1 missense mutation had lower IQ scores, and they also had less efficient cortical brain activity during a working memory task during fMRI. Because of these findings, we think that increasing NAG levels may improve cognition in people with mental illness and potentially other disorders. The fact that we were able to show that higher NAG levels were associated with better cortical activity during working memory and better scores in working memory and visual memory tests lead us to believe that future treatments that could increase NAG levels might be effective at treating cognitive deficits in these patients.